Assalamu alaikum. I would like to welcome you. Again, Assalamu alaikum. I would like to welcome our Prof for being here, giving her precious time to us. I would like to welcome you too. I hope you find this seminar useful. Our seminar is about DNA cloning, which is presented to you by me, Abdullah, Hafsa, and Bensa. After finishing this seminar, you will be able to understand simple informations about DNA structure, then learn how DNA cloning is done. Then, you'll be able to know types of vectors and enzymes that are important for DNA and host cells. Also, you'll know advantage and disadvantage of DNA. Then you'll know applications of DNA cloning.
طبعا اسمح لي اقاطعك، طبعا هذا هو التسلسل الصحيح لاعطاء المحاضره، احنا متجاوزين موجوده يعني متجاوزين، ليش؟ لانه تاخذ شويه من الوقت، بس هو هذا المفروض انه اللكتشر او السيمينار نعطي الاوبجكتيفز، ليش احنا نسوي؟ شنو الفايده منه؟ فهذه الكونستراكشنز اللي هي الانستراكشنز تعطيكم يعني معرفه مسبقه انه على شنو المواضيع اللي حيحكيها.
Before knowing the DNA cloning, I would like to give you some basic information about DNA structure so you'll know the DNA cloning better. DNA word stands for deoxyribonucleic acid. What is the definition of DNA? DNA is the molecule that contains the genetic code of organisms. From the definition, we can know that DNA has two important functions. One is protein synthesis, and the second is holding heritable traits. Someone wants to know what is the structure of DNA. DNA has two strands, two molecules arranged into a ladder-like, which is called double helix. Molecule of DNA is made of billions of tiny subunits, which is called this nucleotides. So once said, what is nucleotides? Nucleotides consist of three main molecules, which is our phosphate group, is represented by a circle, and pentose sugar nitrogen base. And the phosphate sugar and the phosphate and the pentose sugar form the backbone, as you see here. But the nitrogen bases forms the runs. Nucleotides, we have four main nitrogen bases, which is the adenine, cytosine, guanine, thymine. Each base will bond with one another or other specific base. Adenine with thymine, cytosine with guanine. Because of this complementary base pairing in the order of the base in one strand determines the order of the second strand. If we have, like you see here, in the first strand we have thymine, the second strand we will have adenine. In the first strand we have cytosine, in the second strand we have guanine. So it's complementary to each other. I forgot to mention to you that this strand is antiparallel. How is antiparallel? Antiparallel in the beginning we have the 5' end, which has the phosphate group. In the second strand we have third group, which has the hydroxyl group. So they are antiparallel to each other. That is an important note. Now we'll move to the interesting part, which is the DNA cloning. Someone knows what is the DNA cloning? What is the definition of DNA cloning? The use of DNA manipulation procedure to produce multiple copies of single genes or segments. As you see here, we have the DNA strand. We would like to copy only this segment or that gene to make multiple copies of that genes only. I would like to give you a cartoonist story so you can understand how DNA is a clone. We have 5 main characters, which is the restriction enzymes represented by SO, and the DNA, which is represented by DOR, then DNA ligase, which is represented by a glue, vector, represented by CAR, and then host cell, which is represented by garage. our story is then to cut the DNA, cut the door by the cell. Then we use the glue, which is the DNA ligase, and stick it to the car. Then we move the car with the DNA, with the door, piece of door, into the garage. So we made DNA cloning. I hope that you find that useful for you. Now we need more information about DNA cloning, about the procedure of DNA cloning. First, we take the DNA, extract it from bacteria or any other organism, and then we put it in the test tube here. Then we use the restriction enzyme, as I mentioned before, which is a saw or scissor, and then cut the DNA into pieces, as you see here. Only the red one represents the gene of interest. Then we take the plasmids from the bacteria, extract it from the bacteria, and then we put it into another test tube, and then we use the same restriction enzyme to cut the plasmids into pieces, into two pieces. Why we use the same restriction enzyme? That is an important question. Anybody has any idea? Okay. Because of the cut plasmids, that we cut the cut plasmids and the gene size pieces will fit together. They will have the same end, so they will join to each other. If we use another restriction enzyme, there will be no connection between them. That will be fully explained to you by Abdullah then. Then what we do, we will cut the plasmids, we got the cut plasmids and the gene size pieces, and then we mix them together in one test tube. Then we join them together to form recombinant plasmids by an enzyme which is called DNA ligase. As I mentioned before, this is a glue. There is a small note. Some of the cut plasmids that we cut in the restriction enzyme will not join with the gene size pieces. Instead, they will join with each other by DNA ligase. So not all of them will be recombinant plasmids. They will return back as a plasmid. Then, we get bacteria, and they move it to the mixer tube of recombinant plasmids. Then we need to move the recombinant plasmids and then put it into the bacteria by a process which is called transformation. But how this transformation is done? There are many ways to do. But one of them is a sudden change in temperature from 0 Celsius to 42 Celsius, or exposing this tube into quick pulses of electricity to allow the recombinant plasmids to move into the bacteria. So the process is called transformation. Another note, important note, not every bacterium will take one of the recombinant plasmids and become transgenic. So you should pay attention to it.
بس برجع للسلايد. هذه يعني هذه البروسيجر مهمة جدا اللي هو كيف يدخل البلازما الى البكتيريا. البكتيريا ما لها قابلية على الفاجوسايتوزيس، يعني تيجي تلتهب بالبلازما. فلازم انه الذي في الطريقة ينفذ من خلال السيل ممبرين. فاما في الهيت شوك مثل ما قلت لكم عبد الله انه هل الفاريشن يصير بالتمبرتشر. اما هاي تمبرتشر وينزل الى الكولد، حيتكون في البورز ثقوب بداية البكتيريا. هاي الثقوب هي ما راح تقتل البكتيريا، وانما تسمح في الممر حتى تدخل بيد لازمة المولكيول الصغيرة. واضحة؟ او يستخدم انه التيار من كهربائي، فهذا التيار الكهربائي حيخلي ايضا تكون هالبورز صغيرة بجدار البكتيريا ويقدر ينزل من خلالها.
As you know, as we take before, plasmids naturally contain antibiotic resistance gene. In the plasmid we have a gene which is responsible for antibiotic resistance. Therefore, transformed bacteria will have antibiotic resistance. As I mentioned before, some bacteria will not take the recombinant plasmids. So how can we differentiate between the bacteria which did take the recombinant plasmid from those which didn't? So, procedure is called when bacteria are plated. The bacteria are plated into a test tube or a media containing an antibiotic. So, only bacteria which took up the recombinant plasmids will have the ability to survive. Why? Because the plasmid has the ability to resist the antibiotic, and those which didn't take will simply die. Last but not least, the bacteria which contain the gene of interest. As I told you, we need only the gene of the bacteria which contains the gene of interest. So we will separate the bacteria which has the gene of interest from the others, and then we let it divide and make colonies and hundreds of copies. I have a small video so you can understand it better. Firstly, we extract the DNA from bacteria. The desired, we had the red market, the desired gene of interest. We use the restriction enzyme to cut it. We shall have sticky ends. Will be fully explained to you by Abdullah. He is called the sticky ends. And the DNA will be cut into pieces. Only one of them contains gene of interest. And it is the sticky ends. Then we got the plasmids, use the same restriction enzyme to have the same sticky ends. Then, which is the recessor, as I mentioned, then the gene of interest is going to connect to the plasmids by an enzyme which is called DNA ligase, which is a glue, as I mentioned before. So we will have the recombinant plasmids. As you see here, we have a lot of recombinant plasmids, but only one of them contains a gene of interest. So we'll separate that bacteria which contains a gene of interest from those, and then let it divide, and can have the applications of DNA from it. Thank you. I hope you find useful.
بس أعقب على موضوع اللي هو الاختيار الريستركشن انزايم. كل ريستركشن انزايم لها سيكونس يقطعها معين. فإحنا فاللي يسوون الكومبوننت دي ان اي لازم يعرفون السيكونس مالت الجين. لأنه لو يستخدمون أي ريستركشن انزايم ممكن يقطع غير الجين. اوكي؟ فيعرفون السيكونس مالت الجين ويختارون على اساس الريستركشن انزايم. يعني فور اكزامبل هنا مستخدمين الايكو ار 1 هذا حيقطع ما بين الجي والتي. زين؟ فهو يسوي شلون يسوي مثل السكاننج على الجين. زين؟ فوين يلقي وين يقدر يقطع بين السي والجي، فلازم يعرفون السيكونس مالت الجين حتى يستخدمون الانزيم اللي يقطع هذه السيكونس مالت الجين، ولا راح يقطع غير الجين. زين؟ فاذا العمليه كلها يعني معروفه ومدروسه، يعني مو اي انستركشن انزيم. واضحه؟
ثانك يو.
طبعا مرة ما خلص محمود ووضح لكم الستبس مالته.